Hello, my name is Prathamesh Guru, and today I am making a video on technical subject pharmacovigilance topic drug discovery and development. Firstly, we see what is drug discovery. Drug discovery is the process of identifying, designing, developing new pharmaceutical compound to treat, prevent, or any disease. In a past, most of drugs are identified. Most of drugs are discovered either by identifying the ingredients from ingredients from the traditional remedies, and we know the disease are mm, prevent at molecular and physiological state. Then we discuss about the history of drugs. Uh, Firstly, in 1999, herbal drugs was discovered. In 1920 and 30, vitamins and vaccines are discovered. In 1940, R&D boosted due to World War II. R&D means research and development. In 1950, discovery of DNA. DNA was discovered in 1915. In 1970, rise in biotechnology. In 1980, commercialization of drug discovery. And in 1919, robotics and automation. Then we see the drug discovery and development timeline. Firstly, we have firstly in drug discovery we have 10,000 compounds, and after research we got only 250 compounds in a preclinical trial. Then clinical trial we have only 5 compounds, and after a clinical trial there is a one compound which is approval by FDA, and this timeline take 15 to 20 years. Then we discuss about the drug discovery stages. There are 4 main stages of drug discovery. First one is target identification, second one is target validation, third one is lead identification, and fourth one is lead optimization. Also, there are 2 main stages for drug developments. First one is preclinical trials and second is clinical trials. Then we discuss on the target identification. Target identification means identifying and validating a biological molecule. Usually the molecules like protein, enzymes, receptors, and they play a very critical role in a disease pathway. It can be achieve therapeutic effect. Then second step is target validation. Experiments are conducted to confirm that modulating target will have a therapeutic use. Means we did experiment for confirm the modulating confirm the target will have a therapeutic use. Third one is lead identification. Lead identification means selection and finding of a lead compound. They can be easily obtained by naturally or synthetically. Then we discuss on lead optimization. Lead optimization is the process in which optimize the lead activity to improve its target selectivity, metabolic stability, and also potency. Then we see drug development. Drug development means the discovered drug was take into the market is known as a drug development. It consists of mainly 5 parts. First one is preclinical studies, second one is regulatory approval, third one is clinical trial, fourth one is regular approval or in preclinic trials, and last one is post marketing surveillance. Then we discuss on clinical trials. Clinical trials have multiple phases like phase 0, phase 1, phase 2, phase 3. It has a four clinical trial phases, but we say there are 5 phases. There are not 5 phases. There are only 4 phases: phase 0, phase 1, phase 2, and phase 3. Phase 4 is also known as pharmacovigilance. It means post-marketing surveillance. Surveillance means the keep a watch or observation on a drug. And phase 0 is known as microdosing. Phase 1 is known as safety, phase 2 is known as efficacy, and phase 3 is known as the confirmation of efficacy and safety. Then we discuss on the phase 0. Phase 0 is also known as human microdosing studies. First in human trials conducted to study explorer investigational new drug designed to speed up development. Then we discuss on the phase 1. Phase 1 is also known as clinical pharmacological evaluation, first stage of testing human subjects. There are mainly 20 to 50 healthy volunteers. There are three main types of phase 1. First one is SAD, second one is MAD, and third one is food effect. SAD means single ascending dose. MAD means multiple ascending dose. Then we discuss on the phase second. Phase second is known as controlled clinical evaluation. In this phase, there are 50 to 300 patients, healthy patients, and they concerned with safety, efficacy, drug toxicity. Then we discuss on the phase 3. Phase 3 is also known as extended clinical trials. Most expensive and time consuming phase is phase 3, and there are 250 to 1000 healthy volunteers, and this phase is concerned with safety, efficacy, and comparison. And then we know then phase 4 is also known as post-marketing surveillance in pharmacovigilance. They study on adverse drug reaction, designed to detect any rare long-term adverse effect. Then we discuss on the preclinical trials. Preclinical trials are mainly experiments on animals like rat, rabbit. And there are two main types in preclinical trials. First one is in vitro studies. Vitro studies are studies using organisms, that is test tube, using organism experiment, and the experiments are these experiment in test tubes. For example, cell derived from multicellular organism, and molecules like DNA and RNA. There are some advantages like studies can be covered in a short period of time, reduce risk. Then we discuss on the vivo studies. Vivo studies are the experiments using a whole living organism. Vivo study means they use a whole living organism to experiment. It gives vivo studies gives information about metabolic profile, toxicology, and drug intensity, drug efficacy. These are main two stages of preclinical trials. Thank you.